All right, we're going to move on to the digestive system. It is a rather complicated system if you're not familiar with it. I would suggest starting out by learning the anatomy, and that will help you to relate that to the endocrine and nervous system, and then the associated glandular components that aid in the mechanical chemical digestion of food and then the absorption of those nutrients. So we'll go through. I've also included a lot of slides in here that are summary slides that I won't spend a lot of time discussing, but they are designed to help you in your studying and in your understanding of the material. And then this first slide, kind of the red highlighted areas, indicate those parts of the digestive tract that are important when we're talking about the actual movement of the food and digestion and absorption of the food. All right, so let's get started. This is a modified diagram that I use in my animal phys class, and it just, for me, helps to kind of put the overall discussion of the digestive system into perspective. So your body needs a nutrient pool. You need these nutrients in order to continue breaking down and building up and turning over tissue and functioning, muscle contraction and things like that. And so that pool is provided by the food that you ingest. And the reasons behind why you need to eat get into physics and the breakdown of energy and things like that. But we're really focusing in this lecture on this nutrient pool. So in order to obtain that food, you have to eat it, you have to feed, and so that requires sensory systems, the nervous, muscular, and endocrine input in order to allow for that feeding to occur. And we're not going to discuss that very much, but we are going to get into the actual digestion and absorption of that food. And there are two areas of digestion: mechanical, which is the actual physical breakdown of that food into smaller units, and into, you could kind of think of it as breaking it down to the cellular level, and then the chemical digestion, which would be breaking those cells down in terms of chemical breakage of bonds. So actually breaking the bonds that attach chemicals within a cell. Through that chemical digestion, you are then able to absorb. And so you do need that enzymatic digestion of material in order to have it be small enough, to have those nutrients be small enough to be absorbed. And there are various mechanisms through which our body absorbs food, straight diffusion, but usually we're talking about transporters and co-transporters that are allowing for the movement of the food across the membrane. And we'll talk about absorption towards the end of the lecture. Throughout this, we have negative feedback. I'll give a few examples of that, but I'm not going to get into the complete detail about that negative feedback. So this is a good overview of the alimentary tract, or the GI tract. And you've got your oral cavity, or your mouth, the pharynx, which is the region where the respiratory system and the digestive system overlap, and so there is a very complicated series of events that need to occur in swallowing to keep air from going into the digestive tract and food from going into the respiratory tract. So the pharynx is a very important part of this, and we'll talk about that when we talk about swallowing. The esophagus is the muscular tube that transports the food down to the stomach. We generally refer to the stomach as the acidic compartment, because that's, of course, where acid is secreted to allow for the killing of bacteria and the digestion of proteins. Small intestine, specifically the duodenum, is a very important part of the chemical digestion and absorption of nutrients. Large intestine is very important for some microbial activity and for the absorption of water. Between each of these units, so between the mouth and the esophagus and the stomach, etc., we have sphincters. And those sphincters play a very important role in making sure that the food is digested appropriately, properly, and is ready to be moved on to the next segment. The associated glands, what we call the accessory glands, include the salivary glands, the liver, the gallbladder, and the pancreas, and we'll talk about each of these individually in a bit. All right, so something else in terms of anatomy that you should know is the structure of the wall of the gut. And there is a rather standard structure to this. I've highlighted the lamina muscularis, which is variably present, meaning it's not in every part of the digestive wall. And then at the bottom there, you see tunica adventitia. We only see adventitia at areas where the tube, the gut tube, or the alimentary canal, interacts with connective tissue and is not its own separate organ. And so basically what adventitia is, it's this this area where the outer part of the gut interacts with the connective tissue, and so you can't see a distinct lining between the gut wall and the surrounding connective tissue. We generally see adventitia up in the upper part of the esophagus, where we're going from the oral cavity down into the esophagus, and down at the anus, where we're going from the anus. Throughout most of the gut wall, we're going to have tunica serosa, which is that thin lining of epithelium that surrounds the outside of the gut and provides a protective barrier as well. Some other things to note: the tunica mucosa, which is that inner lining around the lumen, that is generally composed of 3 sections: the lamina mucosa, which is your actual epithelium, the lamina propria, which is where you find connective tissue, and the blood vessels that run up and into the propria for absorption, and then the muscularis, which is, as it says, it's variably present. We generally will see it in most of the gut wall. Again, we tend not to see it up in the esophagus and then down near the anus. The tunica submucosa is this area below the connective tissue below the tunica mucosa, and that has it's mostly connective tissue. We also tend to see some glands extending down into there, which are shown. And then we also will see a Meissner's plexus, which is basically the nervous tissue associated with the gut, and we'll talk about that. Tunica muscularis has a very distinct organization. On the inner side, you have the circular muscle, then you have the myenteric plexus separating the circular muscle and the longitudinal muscle. And so we'll talk about why that is such a distinct organization in a bit. So again, it will help you in the previous slide and this slide if you really understand the different sections of the digestive tract and the organization of the gut wall. All right, so now let's talk about hormones. And there is a good breakdown in this provided by your book of an overview of the hormones. And we're only going to talk about hormones. There are other peptides that are discussed, which we will briefly perhaps touch on in a few examples, but we're not going to get into too much detail because it does get very complicated. So gastrin's associated with gastric acid secretion. And so that would mean that it would affect the production of that acid in the stomach. So we generally will see it released from the lower part of the stomach, known as the antrum, and then the duodenum. And there are specific endocrine cells called G cells in the digestive tract. If you took the class with me previously, or you may have heard this term before, we refer to these generally as enteroendocrine cells. So it will cause the release of gastric acid to allow for protein digestion. If it's inhibited, that it's inhibited by acidification of the antrum, meaning once you have enough acid for protein degradation, then you're not going to need it anymore. And it also allows for the growth of the mucosa in the stomach and the duodenum, likely for protection from the acid, because those mucosa will, especially in the stomach, provide that protection. All right, CCK is the big, usually covered in classes, a hormone. It is really important in terms of enzymatic digestion because it triggers the release of enzymes and bile from the gallbladder and the pancreatic exocrine pancreas, which is what secretes the enzymes. It also will secrete some bicarbonate secretion. Again, we will talk about the pancreas in a bit. It is associated with, again, this digestion, this chemical digestion of the mechanically digested food that is leaving the stomach and entering into the duodenum. So we will see it, inhibiting gastric emptying, meaning once there's enough stimulation of it, it will decrease gastric emptying to give time for the enzymes to have their effect. And you can see the stimuli for release there. Generally, it's going to be the foods that are going to be digested, the nutrients that are going to be digested by the effects of CCK. And it will cause increased pancreas and gallbladder mucosa, again, likely associated with the increased need for these enzymes. And this is a diagram highlighting the entrance of these nutrients from the stomach into the duodenum, into the first part of the small intestine, to cause the positive effects in the liver and the pancreas. And so we'll see feedback mechanisms through this. So with increased entry from the stomach, you get increased CCK. That is going to have that feedback back to the stomach to decrease gastric secretion. And so that is one great example of feedback. Okay, secretin is, again, secreted from S cells, another type of enteroendocrine cell. It's associated with the duodenum, and it is stimulated with increased acid. So when you get that input from the stomach, it's going to cause decreased pH, which will cause secretin to be secreted. And so secretin feeds back to the stomach to inhibit gastric acid secretion. As it says, it's basically acting as an antacid to help protect the duodenum from this acid that is secreted in the stomach. So again, a feedback mechanism. When food enters the stomach, it's mechanically digested, it's small enough and ready to be secreted into the duodenum. This will cause the secretion of secretin from S cells, which will feed back to the stomach to help with the neutralizing of the acid. It also, as you can see, stimulates the secretion of bicarb, which is also a neutralizing effect on that acid. And it will cause an increase in exocrine pancreas. All right, GLP, which I actually learned to skip, is in the duodenum and the jejunum. So the jejunum is the first part of, or the middle part of the small intestine. And it's released from other types of enteroendocrine cells called K cells. And it can also be stimulated by glucose release. And it allows for release of insulin to help take up that glucose that has been absorbed, or that needs to be absorbed. And it inhibits, as you can see, gastric acid secretion, another feedback loop back to the stomach. So as the name implies, glucose-dependent insulinotropic peptide, it is dependent on glucose and causes the secretion of insulin to allow for the uptake of glucose, to allow for insulin to cause the uptake of glucose. All right, motilin, other enteroendocrine cells in the duodenum and the jejunum cause the release of this, so M cells. It can also be released through neural control, so neural feedback from the nervous system, central nervous system. It allows for motility, as the name implies, and it causes increased motility through the GI tract to allow for the movement of the food, both to allow for mixing of the nutrients with the enzymes so that they can break down the nutrients, and to keep the bolus or the chyme moving through the digestive tract. All right, this is a great diagram highlighting where these hormones are secreted, and you could perhaps, I would suggest, print out this picture and write in the feedback. Might help to put like little feedback loops in here so that it will help you understand where these hormones are having their effect. The other thing that I want to point out while we're looking at this is, for the most part, these hormones are secreted by the same tissue or within the same system in which they're having an effect. And that's one thing that's really unique and interesting about the digestive system, is that it has its own internal neural and endocrine control and feedback. So even without input from the nervous system, these can all and will all be secreted, and only a few of them will act under nervous system control, like motilin. So that is something to note and be familiar with. And then this is a nice overview of what causes the release either the stimulus or the inhibition of these various hormones. Okay, so we talked about the internal endocrine system of the digestive system. Now I want to talk about the internal nervous system. So, of course, this is known as the enteric nervous system, and we find it throughout the gut wall. I mentioned earlier when we were looking at the anatomy, the myenteric plexus and the plexus in the submucosa. These are part of that. And as this says, they can function independent of the autonomic nervous system, and so sometimes, frequently, this is referred to as the second brain of the body because it is its own nervous system with its own feedback. It's got the same type of cells as the autonomic nervous system, so you've got an entire neuron, cell bodies, axons, dendrites, nerve endings. And it has feedback from sensory neurons, so you'll have afferent neurons causing the contraction or relaxation within the enteric nervous system. And the same types of excitatory and inhibitory transmission that we see in the autonomic nervous system. So again, it is its own system. So this gives a nice overview of the basic principles and anatomy behind the enteric nervous system and the input from the autonomic nervous system. And I put a picture here from McGraw-Hill that shows the submucosal plexus and the myenteric plexus. Now, the myenteric plexus, we'll talk about both of these, but they do have different functions. So the myenteric plexus is important for peristalsis, and the submucosal plexus is important for that, the secretion within the mucosa. And as you probably know already, parasympathetic is generally going to stimulate through the release of acetylcholine, and the sympathetic is generally going to inhibit through the release of norepinephrine. But in the same way that the heart has its own internal pacemaker and control, and then the input from parasympathetic and sympathetic affects that, it's very similar here. So the gut has its own pacemaker cells and its own internal control that can be affected, again, by this extrinsic control. And then this is zooming in on the plexes to give you an idea of how this input works. So as you can see, the myenteric plexus has its own internal structure that communicates with the submucosal plexus, and we've got those sensory neurons. So, for example, you would have chemical chemosensors, touch receptors, stretch receptors, all feeding back into these plexes. So when you eat, if you have an expansion of your gut, that will trigger the contraction within that muscle to allow for secretion and feedback to the central nervous system. And as you can see, the parasympathetic feeds in the same way, and the sympathetic. So if you got release from the parasympathetic, you would have an increase in contraction. If you get release from sympathetic, you would have a decrease. And we will also talk about blood a little bit. A lot of this also has to do with input from the vascular system as well. Okay, so the myenteric plexus extends all the way from the esophagus to the anus. Everywhere where you have peristalsis, you have the myenteric plexus. It's located between the longitudinal and the circular smooth muscle. So it's a pretty standard location, and it is going to cause contractions and movement through the GI tract. With that input from the parasympathetic system, or because of input from the sensory neurons. When you get stimulation, you get increased contractions and increased intensity. So you'll get not only faster contractions, but more muscle will be recruited, so you get increased intensity. The other influence we see is a input into the sphincters to allow for relaxation at the sphincters. Because if you think about it, when you have food in the system and you want to move it from, say, the stomach to the duodenum, you want to have a relaxation of the pyloric sphincter to allow for that movement through. So we do see this feedback from the myenteric plexus into those sphincters to allow for them to relax, to trigger the movement of that food from the different regions, from either the stomach to the small intestine to the large intestine. Or LES is the lower esophageal sphincter, so that would be from through the esophagus down into the stomach. Okay. Submucosal plexus, again, it's extends the same range as the myenteric plexus, and this, as I said, it's different function as the myenteric. It's to help with secretion, absorption, and that in that respect, it causes contraction of that layer of muscle known as the muscularis mucosa. If you look at the structure here, you've got that thin layer of the muscularis mucosa below, in reference to the lumen, so below that layer of lamina propria and mucosa. And so contracting that muscle allows for the glands that are in that mucosa or the lamina to secrete, or to allow for the movement of the cells that are absorbing through the nutrients that are in the lumen, and that will help with absorption. So as I said earlier, it really helps if you know the anatomy well to understand the function of these different plexes. Okay, so I'm going to give one overview example of how the nervous system and the endocrine system work in this in gastric function. And I think your book, it goes through several different examples, and it does talk about this. But I think this is a great diagram and overview from McGraw-Hill to show how the vasculature and the nervous system and the endocrine system work together during filling of the stomach and then movement from the stomach into the duodenum. And we understand a lot about this gastric function, so it's a good one to look at. All right, so there's 3 phases to gastric function, as shown here: cephalic, gastric, and intestinal. You have feedback to your stomach when you're about to eat. So that's what's shown with the vagus nerve. You see food, you smell food, and that causes the beginning of secretion, as it says, even before the food is swallowed. So that positive feedback into the stomach comes from your central nervous system. Then you have your gastric phase. This is when the gastric and the gastrin and the histamine are causing the increased secretion of those enzymes and HCl to allow for the digestion of the food that's coming into the stomach. And this is when food actually stretches the stomach. So that, the middle one here, gastric phase, is internally controlled. You still have feedback from your long reflex, both because you have that stretching, but this is really controlled by that internal ENS and enteroendocrine system. So it's reflexes, as shown here, where you have histamine, so paracrine, and gastrin, that hormone being secreted to stimulate acid and enzyme secretion. Then we have, once the food's been digested enough, so we've got, the stomach is full, we have enough, the food's small enough to move from the stomach into the small intestine, into the duodenum, we've had enough chemical digestion, that's going to cause movement of the food, which, as you can see, triggers the secretion of secretin and CCK. That is going to have negative feedback to the secretion of the gastric enzymes. And so that's why this is a great example of that feedback between the stomach and the small intestine, and it includes several of the hormones that I talked about earlier, which is why I like to give this as an example of the integration between the nervous system and the endocrine system back to the central nervous system. All right, And as it says, this also inhibits that parasympathetic, the vagal input, when we get this release of the secretin and CCK, it triggers that enterogastric reflex, which feeds back to the central nervous system. So we see lots of influence here from central nervous system through specifically the autonomic nervous system, and then internal effects from the internally secreted hormones. And keep in mind, we've got that the myenteric plexus causing the contractions that are going to mix everything together and move the food down and cause it to enter into the small intestine. All right, so as we're talking about the GI tract, it's also important to note it, to discuss the fact that we've got a syncytium here, just like we did with the heart. So we have gap junctions that are allowing the movement of ions back and forth between the cells. This allows the muscle to act as a single unit and allows for variation in contractions because of that, and uniform contractions because of that. So we'll talk about what I mean by variation in a moment. So the syncytium creates this ability for the pacemaker cells to express the signal throughout the tissue, the muscle of the digestive tract, and create what is known as these slow waves. So as it says, that's going to set the maximum frequency of the contraction during these the pace set by the cells of Cajal. And you can get input from hormones and the nervous system to cause changes to that, in the same way that we saw that in the heart. And as this also says, one difference from the heart is that we can also get increased strength of contraction, which would mean bringing in more muscle cells for that contraction to occur. As opposed to the slow waves, we also have these what are called spike potentials. These are action potentials that cause contraction when the slow wave hits threshold. And so that input from the central nervous system, from the parasympathetic system, will create these spikes. This is calcium entry, so it is voltage-gated calcium channels as opposed to the sodium channels. And this is affected by that nervous and hormonal stimuli to, again, increase frequency and cause stronger contractions. But it does not, the maximum frequency is set by those slow waves. So this shows an overview of the input that we can see. So we've got your slow waves coming through, and then when the tissue stretches or you have input from acetylcholine or parasympathetics in general, you're going to have stimulation, and then of course hyperpolarization from norepinephrine or other sympathetic triggers. All right, and finally, the other way that the central nervous system, the sympathetic and parasympathetic system, affect the digestive tract is through blood entry and exit. So when you eat, you're going to need more blood to filter through and take the nutrients out. And so you do see an increase in blood flow for a few hours after eating. And this is triggered by extension, like stretching of the muscle, and other inputs, including low oxygen. So if you think about it, when you've got a lot of activity, you're going to have decreases in oxygen, which is going to cause feedback to increase blood flow. We also see effects from the central nervous system. So the PNS is going to increase blood flow, and the SNS is going to decrease blood flow generally. And you can see an overview of the mesenteric input. And so you can imagine that at these branches you're going to have sphincters, and those are where these inputs will cause effects. So constriction of the sphincters will cause a decreased blood flow, relaxation will cause increased blood flow. All right, so that was the first unit. Now we're going to move through and talk about motility, so the actual movement and breakdown of the food, including mechanical breakdown, so chewing and meaning mastication and deglutition or swallowing. And so this gets into the mechanical breakdown and then the movement of the food or the nutrients through the system. And then later we'll talk about the enzymatic breakdown. Okay, so why do we chew? Why do we masticate? We really want to break the the food down into small enough units that the enzymes can have access. So if you imagine a big piece of starch, even if you have amylase to break down that starch, the amylase is only going to be able to get to the external part. So really decreasing surface area and allowing access to the internal parts of the cells, so breaking the cells apart, is going to allow for the enzymes to function. We also get in mastication, in order to move the food down, we want to mix it with, to lubricate it. So saliva provides that lubrication in addition to the enzymes. So deglutition or swallowing is 3 phases, oral, pharyngeal, and esophageal. Basically, what you're doing, there's 3 different functions in each of these phases. The oral phase is meant to get the food broken down, masticated, small enough, and form what's called a bolus to allow for the food to move down into the esophagus. And so the oral phase is a combination of mechanical processing, feedback, and also analyzing the food, so that's when your taste buds would be affected. And it's when saliva is inserted. So once the food has been masticated and forms what's known as, again, a bolus, then you enter into the pharyngeal phase. Now, the pharyngeal phase is very important. This is when you get this cross-section between the respiratory tract and the digestive tract. And so it is extremely important that you have the correct signals to keep food from going down into the respiratory tract and to keep air from going down into the digestive tract. So there is this coordination between the uvula, the tongue, and the epiglottis to physically block the trachea and allow food to move down through into the esophagus. And once the esophagus once the bolus of food enters the esophagus, then we start peristalsis, and that is going to move the food down into the stomach. We also have the part of this process is relaxing the upper esophageal sphincter so that food can enter through the pharyngeal phase, and then relaxing the lower esophageal sphincter so that food can enter into the stomach. Okay. So this says that the oral phase is also called the voluntary phase, and it gives information about the nervous control. So there's the swallowing center in the brainstem, which takes input from sensory organs in the mouth and the pharynx, and causes the movement and changes to the coordination between the epiglottis and the tongue and the hard palate to allow for that swallowing of the food. So this gives a better breakdown of that and includes the afferent pathways. So you can see that we've got this feedback from all along in the oral cavity and all the way down the pharynx and the esophagus to coordinate stretching of with the bolus of food passing through. You're going to have stretching. That's going to feed back and cause relaxation of muscle and contraction of muscle as you move down the esophagus and through the pharynx. The big input here is the vagus nerve and the trigeminal nerve up in the upper part of the oral phase. All of this is coordinated through what's known as the swallowing center, which was seen in the previous slide. And the other thing that you can see here is this input from the vagus nerve and how it has, again, is going to affect the myenteric plexus and the the myenteric plexus here, really. The other thing to notice here is the transition from striated muscle into smooth muscle. So there is voluntary control over swallowing in the upper, I think it's 1/3 of the esophagus, and then as you move down into the lower 2/3, then you're transitioning to smooth muscle, and that's where you're going to have the autonomic input. Okay. So in esophageal swallowing, we have, again, that primary peristalsis, which is really a continuation from the pharynx, and that is going to go from the striated muscle or the skeletal muscle into the smooth muscle. And because we're going from striated to smooth, we are going to need this feedback of distension or stretching during secondary peristalsis in order to allow for that feedback to keep the bolus moving down the esophagus until it gets into the stomach. Okay, so now let's talk about peristalsis really quick. I think this is a good place. Your book talks about it a little later, but because it starts in the esophagus, this is a great place to talk about it. This is coordination between the circular muscle and the longitudinal muscle of your GI tract, and it is coordinated by that myenteric plexus. As this shows, and this is from a A&P textbook, you've got contraction of the circular muscle keeps the bolus moving forward, and then the longitudinal muscle moves in a kind of a wave and brings the food down the esophagus. So theoretically, if the circular muscle didn't contract behind the bolus, then when that longitudinal muscle contracts, the food could move backwards. So you need this coordinated contraction of the circular and the longitudinal muscle to move the food in the proper direction. And again, this is known as peristalsis. It's this wave-like movement that is created because of this coordination between the two types of muscle. And you need that myenteric plexus in order to coordinate that. Okay. This shows something else that's really interesting. This is the change in pressure that occurs in the esophagus as you move down and as you move from striated to smooth muscle. Notice the effect as well of the diaphragm on this. And we're going to see some really interesting integration between the respiratory system and the digestive system as we're going through. So remember when we talked about breathing, when the diaphragm moves up and down, it's changing the pressure in the thoracic and the abdominal cavity. This affects the pressure in the esophagus and in the stomach, and this can help to move the food down. It also helps with egestion or excretion, which we'll talk about when we get to that point. So the sphincters are an important part here as well. The high pressures associated with the upper esophageal sphincter and the lower esophageal sphincter is going to keep air from going into the esophagus during breathing, and it's going to keep acid from coming up out of the stomach and into the esophagus during processing in the stomach. The manometry is a tool used by doctors to tell if the upper esophageal sphincter and lower esophageal sphincter are working properly, and it all relates again back to that diaphragm and the changes that we see in pressure associated with the pressures in the thoracic and abdominal cavity associated with breathing. So we get an evening out and an elongation of this pressure wave as we get down towards the stomach because of that integration with the diaphragm. So notice that big change in the height of the pressure as we get down into the below where the diaphragm is. Okay, so let's talk about, we're into the stomach now. Let's talk about the different areas of the stomach because it's important. Now we have what's known as the fundus and the antrum. Those are different anatomic regions where there are different secretions and different feedback. We also have something called the orad and the caudad, and this allows for feedback in terms of where the food comes in and where mixing occurs. So we can refer to, again, the ORAD and the CAUDAD as these areas where we either have receiving the food and then mixing of the food. So the ORAD receives the food. And it needs to be able to relax, so you need relaxation of that muscle. And then you need movement of that food down into the caudad so that it can allow for mixing within the caudad. And this, we also see down in the caudad or in the antrum region, we see a lot of the cells that are secreting the HCl and the pepsinogen, that is the enzyme that will allow for digestion to occur. Okay, so again, we see changes in the pressure in these different areas to allow for this movement of food from the caudad region, from the upper part down into the, from the audad down into the caudad. And as this says, peristalsis is an important part of this. So you're going to have these wave-like movements creating this contraction of the muscle that is going to create pressure within these different areas of the stomach. And what this is doing is it's creating, again, changes in pressure so that the food will move down and then churn within the stomach and mix with the enzymes and the HCl. Now, I included a picture in the upper right part of this showing that there's a difference in the stomach in terms of the muscle. We, in addition to longitudinal and circular muscle, we also have oblique muscle. So there are 3 muscle layers in the stomach. This is unique to the stomach. This allows for that churning of the food, and it's important to keep that in mind as we're talking about these pressure changes. We have this additional layer in the stomach that is helping to create the pressure changes and turn that bolus of food into the chyme and allow for that churning to occur. So there is an important regulation that occurs, which I alluded to when we talked about the gastric system earlier. In order for chyme to move into the small intestine, the duodenal part of the small intestine, we need it to be to have a slow movement to allow for neutralization of the pH, because again, you're going to have that really low pH in the stomach entering into the duodenum, and you need it to return to a neutral pH in order for the enzymes to function in the duodenum and to protect the duodenum from that really low pH. Excuse me. Until we get the contractions of the gastric muscle and the relaxation of the pyloric sphincter, we're not going to get emptying. So that pyloric sphincter is very important to regulate the movement of the chyme from the stomach into the duodenum. So how that occurs is the changes in the ORAD and the CAUDAD that occur in the contractions will eventually decrease the pyloric sphincter and cause emptying of the gastric chyme into the duodenum. Okay. And then we get, when there is enough emptying occurring, we need feedback to decrease that entry. So we get when there's relaxation of the ORAD, which would result in decreased contractions, that's going to feed back to that sphincter and increase it, so contract it more. And in addition, as it says here, we've got segmentation occurring in the intestine, which means that the food is forming, the chyme is forming these segments. And that indicates that there's food there, and therefore there should be a decrease in the entry of food from the stomach into the duodenum. So that decreases gastric emptying. So immediately after a meal, we're going to have regular, you could think of it as regular contraction in the pyloric sphincter after digestion has been occurring for a while, then we're going to get relaxation of the sphincter to allow food to move in. Now, the food does have to be a certain size to move through. I think it's something like 2 millimeters or something. So that sphincter is still going to have some control over what's moving into the small intestine, so that it's small enough that the enzymes can then access it. Then receptors are activated during once enough food is moved in and we have segmentation in the small intestine. Then we're going to get restriction again of that pyloric sphincter. So we, as you've probably noticed when we're talking about that, there's a lot of feedback that's occurring. We have regulation based on receptors that regulate based on the osmolarity, how much acid, fat, or protein there is relative to the nutrients that are being digested. So what triggers this is CCK, which we talked about earlier, and again, that's triggered by fats and proteins. And if there's too much acid, that's going to restrict the gastric emptying because, again, we need to have a neutral pH in order to have appropriate digestion occurring in the duodenum. So now we're in the small intestine. So we've moved through the esophagus, the stomach. We're now in the small intestine. The purpose here is to move the food through, mix it together, more so than in addition to what occurred in the stomach, allow for the access of the enzymes, which came from the pancreas, to the food. And so we have had most of our mechanical breakdown occurring to this point, and now we're going to have chemical breakdown occurring. So that's the primary purpose of this motility, is to mix. We also need to neutralize the pH, and so the mixing of the chyme allows for that. So we have an input of bicarbonate, which is going to help neutralize. Then, once the chyme's been mixed and also exposed to the mucosa to allow for absorption, we also need to move it through. So we've got, that's why we have what's called segmentation in addition to peristalsis, to help move the food through and allow for it to mix. So that's what this is showing. Peristalsis is going to propulsively move the food through. We already talked about with the esophagus, same things happening in the stomach and in the small intestine. However, because in the small intestine, we also need that food to mix within a certain region and have access to the mucosa for absorption, in a similar way as in the stomach, except we have a lot more absorption occurring here. We need segmentation. And so segmentation is an additional contraction that keeps the food in these regions of the duodenum, small intestine and large intestine, I should say, throughout, to keep it moving. And as you can see here, we can have different spacings, which will all depend on the food, feedback from the afferent neurons, and the insertion of enzymes. So now we've gone through the small intestine. We'll talk about absorption and chemical digestion later, but that has occurred. Now we are going from the small intestine into the large intestine. We encounter another sphincter here, the ileocecal sphincter, because it is connecting the ileum and the cecum. This opening of this sphincter is dependent on pressure and other afferent neurons, so that would cause peristalsis to occur. The contents also have to be fluid enough, in a similar way as with the pyloric sphincter. So we have this control over the food, the chyme going from the small intestine into the large intestine, and again, it's also acts as a valve. So sometimes you'll see this referred to as a valve, because we don't want the food to go from the colon back into the ileum, and so it's a one-way movement with regulation. So as this slide shows, the pressure is what is allowing for the movement of the chyme from the ileum into the cecum, or from the small intestine into the large intestine. So when the ileum is stretched or distended, that will cause the relaxation of that sphincter, and the pressure changes that you can see here will allow for the food, the chyme, to move from the small intestine into the large intestine. When there isn't, when the colon is distended, so when the cecum or the colon is distended, that is going to change the pressure and constrict the sphincter. So again, this is going to allow for chyme to move from the small intestine to the large intestine when there's space available, basically is what this is showing. So the large intestine is really important for, again, mixing chyme. We do have microbial digestion going on, especially in the cecum, and so there are some additional nutrients, specifically some vitamins, that are being able to be accessed in the large intestine. But in general, we are absorbing electrolytes, absorbing a lot of water, and propelling the fecal material through what's called mass movements. So, as it says, the large intestine is extremely important for storage and for creating the solid material that will be expelled, and then storing that solid material. So that's why I say there's a lot of water absorption going on here, because there's relatively slow movement of the chyme through here to allow for absorption of that water and then storage of the fecal material. As I mentioned earlier, when you get into the large intestine, the product is moving through what's called mass movements. So this allows for defecation, it eventually, and it produces the mass of fecal material. This is a result of the anatomy of the large intestine, in addition to the feedback mechanisms in terms of the stretching and the relaxation that occurs when the movement is going through. And it's represented here by a barium, which will follow the food through the large intestine, and can show you how you get this kind of larger segmentation, which we call a mass, because it's not just one segment as you move throughout the colon. And there are reflexes involved here. So the gastrocolic reflex and the duodenal cecal reflex will feed back to prepare. So as you can imagine, distension of the stomach, that is preparing the large intestine for food that will get there in several hours. And then of course the duodenal cecal reflex is going to have to do with when food gets into the small intestine. Again, you need preparation in the large intestine for that food to be able to enter. All right, so finally, let's talk about defecation. So again, there's feedback mechanisms here. And as you might guess, it has to do with stretching. So when there is when space is needed and there's an adequate buildup of feces within the rectum, that's going to stretch those receptors, that's going to feed internally and back through the spinal cord. So there is a spinal reflex here that relaxes the internal anal sphincter. So something important here is to note that the internal anal sphincter is smooth muscle and the external anal sphincter is skeletal muscle. So there is a involuntary feedback to relax that internal anal sphincter when there is a bowel movement present, but the external anal sphincter will remain constricted until a voluntary input, and that's going to come from the cerebrum. So you have conscious voluntary control over defecation. And that's aided by these other inputs related to your higher centers in your brain, meaning your cerebrum. One of them is known as the Valsalva maneuver, and so this is taking advantage of those pressure changes that occur when you move your diaphragm up and down. And so when you defecate, you will sometimes use this Valsalva maneuver to create the pressure in the abdominal cavity that helps to push the feces downward and out. And this will also help move other materials through the digestive tract because of the differences in pressure. If there is a transection of the spinal cord, this will affect that reflex and cause problems being able to defecate properly. All right, so I put in check your understanding at this midway point, and again, go through and answer these questions, and then let me know if you have any questions.